We designed primers, 4 primers, primer 1, 2, and then primer 3 and 4, right? And then uh, we had set up the reaction in 2 different tubes. In tube 1, primer 1 and 4, which led to amplification of one half of the gene, and in another tube you had combinations of primer 3 and primer 2, which led to the amplification of the second half of the gene, right? Finally, what you do is that in another tube, which you can label it as tube 3, you add primer 1 and then primer 2, which are forward and reverse primer. And in this case, the template that is used is the template obtained from tube number 1 and tube number 2. That means the 2 halves of the gene, right? So in this case, class, like Ashi was asking in yesterday's class, that the chances of formation of these halves are high, right? But then the chances of formation of this combination, right, the one which is shown in the top diagram, is less. So that is actually very true. More of these fragments will self anneal, will anneal with themselves, but then there will be a minority of population where, due to the overlapping region which is shown in red, we will have such a representative structure formed. Because of this overlap, you can have such a structure formed, right? Now the point is that, class, when we use primer 1 and primer 2, right? So when we use primer 1 and 2, we cannot get enrichment of these halves. We can get only the full length fragment, and that is possible only when we have the extension of this overlap. And therefore, class, this technique of PCR is also called as overlap extension PCR, right? It is also called as overlap extension PCR, because in this case the overlap is being extended, right? This is the overlapping region and this is being extended with the help of TNTPs. So therefore, this is also called as overlap extension PCR. And then we discussed that you can actually remove the template, and which can be removed by digesting it with DPN1 enzyme, which actually digests the methylated DNA. This is how you are enriching the mutated fragment largely, because the non-mutated ones will be removed by the enzyme DPN1, point number 1, and these two halves cannot be amplified. Even if they self-anneal, they cannot be amplified by primer 1 and primer 2. So therefore you will have a vast majority of population which is actually the desired sized fragment which contains the mutated one. Still, if you are worried, you can always check whether you have pure population or you have some population which is wild type. That can be checked by sequencing, by gene cloning, or by, if in case by mutation a unique site is generated, you can subject it to restriction digestion and run it on a polyacrylamide gel, because the concentration will be too less. So if you load it on an agarose gel, it will not be visible. So therefore you will have to run a polyacrylamide gel. Then we were discussing the PCR program. So in PCR program, we have the cycles. So we are going to do hot start. So that means you will have a step of denaturation. Denaturation is usually done for maybe 30 seconds to 1 minute maximum. Only in certain conditions which you will be actually doing it in your labs. So when the template is, I'll write it, so when the template is pure DNA, right? Pure DNA, like it can be pure genomic DNA or pure plasmid DNA. So in this case, the time is 30 seconds to 1 minute. Template is a pure colony, right? Pure bacterial colony. And in this case, you will have to take cells. So here, these are bacterial colony or bacterial cells which are used as templates. So in this case, you need to incubate for 10 minutes at 95 degrees. Okay. Following or no? Why 10 minutes?
Yes, ma'am.
Because you need to lyse the cells, because the template is now inside the bacterial cell. So when we heat it at 95 degrees for 10 minutes, usually gram negative bacteria easily lyse. For gram positive bacteria, we may have to do wall treatment as well and then subject it to this kind of lysis. So this is just done for 10 minutes. After that you can have the cycles, which can be, you can do up to 30 cycles, 25 to 30 cycles. So denaturation 95 degrees for 30 seconds. Annealing temperature is usually melting temperature minus 5 degrees Celsius. So you calculate the melting temperature and then usually so they say that you can take 3 to 5, but in general people go by 5. And then this again is done, and extension is done at 72 degrees. And why 72 degrees? Optimal temperature for the polymerase, optimal temperature for extension of the polymerase. And in this case it varies. So for example, for Taq polymerase it is 1 kb per minute. You remember that?
Yes, ma'am.
Finally, so you repeat 30 cycles of this, and
Ma'am.
finally we keep one final extension, and this is done for 10 minutes, 7 to 10 minutes. And the reason we do this step is to ensure that any templates which are truncated, which are not extended, will also be extended in this step. And finally you keep it on hold at 4 degrees Celsius, and then you can leave this overnight, or depending upon the time you can keep it for 3 hours, 4 hours, as per your requirement. So now the point that I was trying to tell you is that when we are doing site directed mutagenesis, the PCR program will be exactly same, 30 cycles, 30 cycles for each amplification. Everything is same. Only thing is you are not going to use Taq polymerase, right? Because it is site directed mutagenesis. So rest of the protocol would remain same, but instead of use titanium Taq, or you can use fusion Taq, any high fidelity enzyme would do. Okay, and then
Ma'am,
yes,
why do we have to do only 30 cycle? Can't we do like 40 cycles or 50 cycles if we want like more polymerase, more number of copies?
yes, you can, but then the number of the more you increase the number of cycles, so you will have to. So it is a good idea to scale up the things, because usually the protocol is set in such a way that you have enough primer, enough template, enough DNTPs, and the polymerases. So all of these things are usually commercially available. So for example, we buy polymerase from New England Biolabs, right? NEB is the name of the company, New England Biolabs, or you buy from In Vitrogen. So they come with a standard protocol. So they recommend that, okay, for In Vitrogen, for platinum reaction volume to up to 50 microliter, something like this. Likewise for NEB, they said that the final volume should be 30 microliters, and then this is the standard. So if you want to scale up, I guess everything has to be scaled up. And then I think the more number of cycles you do, the chances of error would increase, even with high fidelity enzymes. So that is why it is not recommended, because if you see the papers, you will find that when they claim that this is a high fidelity enzyme, so they say that it has proofreading activity, it is a high fidelity enzyme, but then for that the comparison is with Taq polymerase. Nobody says that it is 100% proofreading. They only say that, for example, 250 times better than Taq polymerase, or 500 times better than Taq polymerase, something like this. So I think if you increase the number of cycles, the chances of error could be more. Okay. Then another question which was raised in yesterday's class was mutation in non-coding DNA. So mutation in non-coding DNA can also be induced, because non-coding DNA also at times is important, because non-coding DNA can be not only repetitive DNA, but non-coding DNA can be coding for regulatory regions. So for example, so this you will learn in detail when I will be teaching you transcription. But then for example, the transcription has to be terminated. So there are sequences called terminator sequences required for the binding of RNA polymerase for initiation of transcription. So in principle class, these are non-coding, right? They are not coding for any gene, but then they do are important. So any mutation, sometimes we induce mutation in the promoter DNA to enhance the activity of the promoter, so that the number of transcripts increase, and therefore you will have more amount of protein, something like this. But we will discuss this in more detail in the later classes when I will be teaching you transcription. But then just to answer this question, that yes, mutation is sometimes induced in non-coding DNA as well. Okay, class, are you people there?
Yes, ma'am.
Okay, is it
Yes,
clear?
ma'am.
Yes.
Okay, so then so this is overlap extension PCR. At times another variant of PCR is called nested PCR. So nested PCR is done when sometimes the amplification is very weak, the amplification is less. So in this case what is done is that you perform first PCR reaction. For example, you select a primer which binds at a faraway distance, and then use this as template and design internal primers, right? So you get an amplicon after first PCR, and then you use that amplicon as the template and then use internal primers. Look at the difference, right? Difference between the position of the first primer and the first primer which is used in first PCR versus the difference in position in the second set of primers. So these primers are called internal primers, and they will bind in a sequence which is internal to the sequence which is generated by the first amplicon. So after second PCR you will get enough intensity. So this is used for improving the specificity, or sometimes when the yield is less, sometimes when you have non-specific bands. So in such cases we use PCR product as template and perform second PCR, and this PCR is called as nested PCR. Okay. Now PCR can also be used for incorporating new sequences, and now slowly we will move to gene cloning. But maybe before that I would like to tell you that PCR can be used for various techniques. For example, PCR can be used DNA fingerprinting as well. The question is how PCR can be used in DNA fingerprinting. So PCR is used in DNA fingerprinting class. So what happens is that particularly when you are trying to solve a murder mystery, or when you are trying to solve a crime case, right? So usually when we talk about the DNA, the amount of DNA is in the form of a spot of blood, right? Just a spot of blood, or maybe just one hair, one strand of hair, right? So my point is that the point I'm trying to tell you is that the amount of DNA may not be enough for restriction digestion. You are getting my point? So what we had discussed earlier was that we can do RFLP, and then we can do southern blotting, and then we can do fingerprinting. We can design a probe which is specific to the repetitive sequences, right? But then that method will work when you have sufficient amount of DNA. But usually when it is a criminal, when it's a crime case, then the amount of DNA is usually very less. Sometimes you will find a spot of blood or a little bit of saliva or a little strand of hair. So in such cases, the amount of DNA that you will get from a single strand of hair or a spot of blood may not be enough. Are you people following, class? So in such
Yes,
cases,
ma'am.
okay, so in such cases, class, what is done is that we take the help of PCR. So in this case, primers are designed based upon the conserved sequences. And you know that, maybe I will tell you this first. So PCR, class, I think I have discussed this before, but in case no, then I will tell you one more, that for PCR and using Taq polymerase, you can easily go up to 2 kb, and then maximum you can go up to 5 kb. Beyond that, it will not work, right? For certain other high fidelity polymerases, I will write high fidelity polymerases which have high processivity as well. So in such cases, you can go maximum up to 6 kb in real life. But there are certain other polymerases which are called as range polymerases. Using these long range polymerases, you can go up to 10 to 15. That's what they claim, but I haven't seen anybody going beyond 10 kb usually. So the reason I brought it up here is that with PCR, you can amplify easily maximum up to 10 kb and not more. Okay, so in this case, when you have a spot of blood or when you have a strand of hair, so what I was trying to tell you is that the amount of genomic DNA may not be enough for RFLP. So therefore, the other choice is to use primers and then perform PCR. So in this case, primers are designed based upon the conserved sequences and repetitive sequences, which are in this case called tandem repeats. So this VNTR region can be amplified. You can go easily up to 10 to 15 kb using long range polymerases, right? Otherwise, up to 6 kb without any trouble. So once you have this amplification of the VNTR region, class, then what you can do is you can subject it to restriction digestion, and in this case, class, no need to do southern blotting. Why do you think southern blotting is not required now? I hope you can answer this question very easily. Class, what happened? Are you people there or left?
Ma'am, we are here.
Okay, let me see. I'll open Teams. So, Jaskin, Udita.
Yes, ma'am.
Yes, ma'am.
So tell me, why do you think southern blotting is not required, or do you think southern blotting will be required? Is the idea clear?
Yes, ma'am. But in RFLP also we do not do southern blotting.
What about others? Dhwani, Vishantan, Udita, Jaskin, Gopika, Narendra, Bhavya. Say something. Paras. Arey, what happened to my class? Ashi?
Ma'am, I guess because we are designing primers for conserved sequence, so we already know it will amplify some specific part of the DNA. So we already know that what they are. So we
Very good,
just
Deepak. That's
try
the
to
answer.
start.
So why you people are taking so long? So in RFLP, Ragnesh, if you remember, we discussed that whenever, right? So whenever your starting material is genomic DNA, so when you perform restriction digestion, you will have multiple fragments. So RFLP alone without southern blotting will not give you the correct number of repeats. You remember I had shown you with the figure.
Ma'am, but why do have to do blotting process?
Otherwise, how will you tell me how can we identify the number of repeats? Let's say you have 2 repeats, Paras has 3, Udita has 4. How to determine that? When I will isolate the DNA from the 3 samples, then how will we know? It will give me a mere 3 billion base pairs. RFLP is restriction fragment length polymorphism, right? So how am I going to identify the band which is coming from VNTR unless I do southern, which is specific to the
Yes, ma'am. I understood.
followed or no?
Yes, ma'am. Yes,
What
ma'am. Understood.
others? Dhvani, Gopika, Vishantan, Ashi?
Ma'am, it is clear.
Yes, ma'am.
Yes, ma'am.
Okay. So then my point is, so in this case, now everybody agrees we will not require southern blotting because you are starting with a very specific amplicon, and then you can simply digest it. And since it's a 10 kV region, what kind of electrophoresis we will do? Let me see whether my class is following. What kind of electrophoresis we will do to separate?
Electro.
Hmm? Deepak? Rajneesh, you were saying something?
Ma'am, agar वो gel electrophoresis.
Gel electrophoresis. Right? Everybody in
ये
class?
ये
Huh?
मैम PFGE और agar वो gel electrophoresis.
No, simply agarose gel. Why you will do PFGE? PFGE is for larger fragments. In this case, it's a PCR product. So is it confusing why everybody is quiet? In yesterday's Viva, some of the students told me that, ma'am, some of the electrophoresis techniques are confusing. Are they confusing, class? Please ask me if you have any doubts. So it's easy to remember whenever it's polyacrylamide gel electrophoresis, it's going to be small oligos, only maximum up to 500 base pairs, not more, right? When it is PFGE, it is for larger genomes in megabases, right? PFGE is in oligos, base pairs, small oligos, 100 to 500 base pairs. PFGE pulse field gel electrophoresis is for megabases, genomes, large genomes, right? Agarose gel electrophoresis is for smaller plasmids. When we talk about megaplasmids, which are in 1 MB or something, megabase pairs, then again you can use PFGE. But for small plasmids, you can use agarose gel electrophoresis, right? So in this case, because the amplicon is only 10 to 15 kb, so in this case we can use simple agarose gel electrophoresis and you will be able to identify the differences. So PCR can also be used therefore in DNA fingerprinting. PCR can also be used for designing a probe. So class, don't sleep. Then nobody's speaking. Are you people there?
Yes.
Okay. So what is a probe? Tell me. Narendra, what is a probe? Gopika, would you like to try? Paras, Dhwani, Jaskin.
Ma'am, a probe is basically a sequence that we can use for identification. So, I mean, a small sequence that we can hybridize with our maybe gene of interest or something.
Right,
to
very good. So a
identify.
probe has usually a fluorescent tag or a radioactive tag, right? So PCR can be used for the preparation of probe because probe sequence as large as up to 500 base pairs, and if you remember we discussed in one of the classes that you can use even whole plasmid as a probe. In such cases, when it is whole plasmid, you can simply isolate the plasmid and use it as a probe. But for example, when it is a gene, for example, the gene for green fluorescence protein, right? For certain enzyme, for example, alpha amylase, you want to pick up alpha amylase. Possibility is if the gene of GFP or alpha amylase is there in some plasmid, you can simply perform restriction digestion, obtain the fragment, gelilute the fragment, which I have already taught you, and then label the fragment with a radioactive isotope or fluorescent tag and use that as a probe. But just in case, if that clone or if that plasmid containing GFP or amylase is not available, then you can primers and then amplify the gene for FP or amylase and then use it as a probe, right? Followed or no?
Yes, ma'am.
Okay. Okay, so then PCR can also be used for incorporation of new sequences, and here I would like to introduce you to the next topic, which is gene cloning. So PCR can be used for cloning a gene. This is exactly the same sequence for which we had designed the PCR primer. So I'm taking the same sequence. So in this case, it is a coding DNA because it is starting with ATG and ending with TGA. So then how PCR can be used for gene cloning? That's the question. And then the next question is why how PCR can be used for gene cloning, and then how restriction enzymes can be used for gene cloning. But let's first understand why we should do gene cloning. What's the need of doing this process? Why do we need to clone a gene? Okay, tell me, class. Udita, so you people have some quiz or something? Why everyone is quiet today?
No, ma'am, we don't have quiz, I think.
Then what happened?
Ma'am, we need to clone genes to obtain several copies so that we can perform like multiple experiments. Or
Yes, ma'am, perhaps gene cloning we have learned a bit.
Okay, so then tell me. Then you should
ma'am,
answer these
random
questions
mutagenesis.
fast. Hmm?
To have random mutations.
Random mutations. Okay. And
Gene dosage.
gene dosage. Okay. Dosage and copies are more or less same, but it's okay. I've written that.
Like to supply that gene in the heterologous host.
Okay. So have you studied already PCR, site-directed mutagenesis, gene cloning?
No, ma'am.
He told me that you people have studied. Somebody said na ki gene cloning you have studied.
Yes, ma'am, I said just you have we had a study just here.
Okay, okay. So then tell me, why do we clone a gene? Anything else? Ashi, Udita?
For medical applications,
Okay.
understanding the impact on the genes.
And
Ma'am, more than that, what can be just this type of things like for experiments? So we have more copies, and more than that, what can be?
what?
I said that just gene cloning is for obtaining several copies, and all the things are there.
That's it?
Ma'am, I
Yes.
guess gene,
Hmm?
ma'am, I guess gene is used in industry to produce insulin from bacteria because they just take the gene of insulin and insert into in some another kind of microorganism and then produce it in large amount and then extract the insulin for
Okay.
commercial use.
Commercial purposes, yes.
Ma'am, but for that purpose, we need just a few copies of gene, and then we will, once we insert it into bacteria, then we can use that bacteria for and grow. We don't need again and again produce gene cloning and insert that into bacteria.
Right, so the main point is that to avoid, so for example, particularly for eukaryotic systems, so for example, if you are working with a brain tissue or you are working with, for example, a liver cell, right, or you are working with, I do not know, you take any tissue, so in such cases you don't have to every time sacrifice the animal or the tissue, or I do not know, whatever it is, so you don't have to sacrifice. That's the biggest advantage of gene cloning, because once, so if you keep on obtaining the, if you think that you want to obtain, for example, Deepak said insulin, or if you keep obtaining, let's say, the gene for granulocyte colony stimulating factor, or any other gene which is from brain cells or something, then in this case every time if you sacrifice and you do prepare cDNA, and then so that thing is saved, because every time once it is there, whether it's in a mammalian host or whether it's in a yeast system or if it is in a bacterial system, but once you have generated that clone, then it is good forever. You don't have to every time sacrifice the animal, right? That's the biggest advantage. The second thing I wanted to tell you, because from gene cloning we will slowly switch to sequencing. So sometimes we do gene cloning for at times when we want to determine just the sequence, right? So for example, let's say there is a novel bacteria, right, or a novel yeast strain or a novel Saccharomyces strain, and in this case, for example, the sequence is not known, you do not have any idea, but you would like to sequence, for example, you have identified or you are suspecting that this is likely the locus, right? So in such cases, there are two possibilities: sequencing the entire genome, which can be very tedious. So in such cases, what you can do is you can simply clone a gene and then go for sequencing. Sequencing is also used many a times, particularly, so these days next generation sequencing platforms are available, but then shotgun cloning is done for whole genome sequencing. So I'll be teaching you that how gene cloning is useful for sequencing. But let's first start with gene cloning. What is gene cloning and how do we do that? So coming back to this gene, so in this gene we have ATG is the initiation codon and TGA is the termination codon or stop codon, right? So when we say initiation codon, this is for translation initiation of translation, initiation of protein synthesis, and termination codon is for termination of protein synthesis, right? So if you want to clone the gene in this case, what we do is that, so this part you need to listen to me carefully. So if you want, right, by PCR, what we do is that we insert a sequence. For example, so in this case I am taking the example of EcoR1. This is the recognition sequence for EcoR1 class, correct? If you will not speak, I will not understand whether you people are following or not, and then I will keep repeating the same things.
Yes,
Ma'am, it's clear.
Yes,
we are.
ma'am.
In this case, class, what we can do is we can add a site, a sequence, a recognition sequence for EcoR1, and the recognition sequence for EcoR1 is GAATTC. Okay, but when we do this, right? So in this case, we will have to provide a few extra bases as well, and these extra bases can vary. So for example, we add GCAT, something like this, and here also you can add G T or something. This is just a random sequence. Okay, now what is the purpose of this random sequence? Why at all we need this random sequence in the primer for cloning the gene? Purpose of this random sequence is, class, when we do this PCR amplification, the product will be a linear amplicon, correct? So in this case, when we perform the restriction digestion using the enzyme EcoR1, then if we do not add this sequence, right, if we stop here only at GAATTC, then it has been experimentally demonstrated that the digestion will not be 100% efficient. And the reason is because these restriction enzymes, if you remember from the class where we discussed restriction endonucleases, so these restriction enzymes, they exist as dimers, right? And they cleave double-stranded DNA. So when they bind at the ends, they do not bind properly because the site is almost at the end. Remember the palindromic sequence? So in this case, the top strand will be cleaved between G and A, but the bottom strand will be cleaved almost at the edge. There will be just one nucleotide, right? So therefore, if you provide some extra bases which vary from enzyme to enzyme, for EcoR1 it requires 4 nucleotides extra for properly binding and cleaving. For example, for NDE1 it requires only one nucleotide. For SMA1 it again requires only 3 nucleotides. So it has been experimentally demonstrated that you need to provide a few extra bases so that you can have the digestion, proper digestion. Okay, so now class, when we design the PCR primer, is, so when we design the PCR primer, so the primer should have the sequence of both the one which is shown in blue, the one which is shown in red, and then the one which is shown in black, right? So the PCR primer will actually have therefore an extra sequence. Are you getting my point or no? So this sequence is extra.
Oh, yes, we are getting. Yes,
And therefore, in the first cycle, the primer will hang up like this. Because the which needs to be amplified is only this. This part is not which is present in the sequence. This we are introducing artificially for cloning the gene. Is this making sense or no?
oh, yes, ma'am. You are understanding.
Deepa, Kashi, Dhwani?
Yes, ma'am, it is.
Yes, ma'am.
Yes.
Okay. So therefore, when we design the PCR primer, we will have to add these sequences as well. Okay, and therefore these primers become longer in length. So for example, the forward primer will be having this whole sequence, which is extra sequence, followed by EcoR1 site, followed by this. Now the other point that you need to pay attention over here is that, see that the recognition sequence for the restriction enzyme is 6 nucleotide, GAATTC, right? 4 nucleotides you are supplying extra, so then this total is 10, correct? So that means this is the part This is the part which is bind to the template DNA. This is the extra DNA that is introduced in the primer. Yes?
Yes.
Yes, ma'am.
Therefore, so in such cases, if we assume that the primer length has to be only 15, so then it should stop here, because 10 nucleotides are already extra, and we have added now 5 nucleotides, so it is 15, right? So in this case, do you think we will get amplification?
Ma'am, that should be C, ending at C or G, right?
अच्छा ठीक है, okay, I will add a C, but still you will get an amplification.
Ma'am, if primer binds, so we can get.
Hmm?
If
That
primer binds, we can get.
okay, let's see who else wants to try.
And we may not get the amplification of the required gene if we use this primer.
Okay. What about others? Why so?
Ma'am, because the primer could bind at some other site where where it is more homologous, as in the sequence is more similar or complementary.
So why it can bind to other places?
Ma'am, the primer also has a
Because we
sequence. So
have only 6.
Yes,
Sorry.
that's the point, because it has only 6 base pairs. I mean, you have only 6 nucleotides. So the specificity of the primer is not there at all, right? Because this part is not present in the template, right? Only the black sequence is the one which is native template, and this black sequence, the the chances of finding the 6 base pair sequence is going to be after every 4096 base pair that the class remembers. So in this case, class, we cannot, if we just stop here considering the logic of 15 nucleotides, then the primer will not work. So therefore the designing of PCR primers that we had discussed, so please listen to me, this part you need to remember this very carefully. So when we design the PCR primers for just amplification, then all those rules which we had discussed, that the length should be 15 or 15 to 18 or 15 to 20, so those rules are applicable. But when we are specifically interested in cloning a gene using PCR primers, then length is not the criteria, then specificity becomes the criteria. So therefore in this case, class, the length of the PCR primer, you can go as long as, see, the idea is that 10 nucleotides 10 nucleotides are not hybridized with the template. Then what if you have 6, the binding is not going to be good. So the extra nucleotides, the extra black ones, they have to be more than 10. They have to be more than 10. So therefore the length has to be more than 20, and usually it works really well if you have the length of, you can easily go without any trouble. Depending upon the TM and other factors, you can go easily up to 25, 28, 30, 40, sometimes 42 even. Such cases, when you have such long PCR primers, the formula calculating the TM will not work. So in such cases, you can use softwares. So there are primer designing softwares. There are more complicated formula that actually take into the consideration the entropy, the Gibbs free energy, because whenever the length increases, the chances of formation of secondary structure increases. So those things are also taken into consideration when they calculate the melting temperature of longer primers. So you can use softwares, or you can use, there is a bigger equation which can also be used manually to calculate, but the formula that I had discussed in the last class, in the previous classes, this is applicable only and only for smaller primers. Point number one, the primer designing rules are all fine. That means in this case also you are going to end with a G or C at the 3' end. That rule is applicable. You are going to avoid stretches of G's and stretches of A's, T's or C's. But then if they are part of the gene and if they are there in the sequence, nothing can be done. You can only increase the length to enhance the specificity, and that's it. Nothing more can be done. Okay, so when we talk about gene cloning, the rules of length and the calculation of TM changes, but the remaining rules will be applicable. Hope it is clear to the class.
Yes.
Yes, ma'am.
Yes, ma'am.
Likewise, when we design the reverse primer, again you can design the reverse primer using the same idea. So you will have, so the primers are designed from 5' to 3' direction. Also you can design reverse primer. So I think the class knows how to design the reverse primer, so I will not go into the details. So once you have once you have designed these primers forward and reverse, you are likely to get an amplicon, right? And this amplicon will have at both the ends equivalent site. Yes?
Yes, ma'am.
Yes, ma'am.
Okay. So once you have this amplicon, which is in fairly good concentrations, let's say in micrograms, and which has at both the ends equivalent site, we going to do, then what we can do is we can subject this amplicon to restriction digestion with EcoR1, and then it can be cloned in a plasmid. So usually plasmids are used as vectors for cloning. So what are vectors, class, and why plasmids are used as vectors? So you will learn about vectors in a lot of, in a great detail in 7th. Introduce you the topic so that when you will go in the course you will be at least familiar with the terms like what is a vector. So vectors are actually carriers of genes. So vectors are something which can carry the gene, right? And in this case plasmids are used as vectors. But then there are certain other examples. For example, phage. There are viral vectors as well. So phage DNA can also be used as, so these are called as phage vectors. Phage DNA can also carry artificial or foreign DNA, and these vectors are called phage vectors. Then people also use high capacity vectors such as there are megaplasmids. For example, P1 is a megaplasmid as vector. So I'll be telling you what are high capacity vectors and what is P1 in a little more detail, but then just to tell you that vectors are the ones which carry the gene and they are used for gene cloning. Now depending upon the size of the gene, class, depending upon the size of the gene, the choice of the vector may vary. So the choice of vector upon a few factors: A, choice or the size of the gene; B, the host organism; and C, the any modifications. So what I mean is that the choice of vector depends upon the size of the gene, in the sense that when your gene is like really small, then you can simply go for plasmids as plasmid vectors. When your gene is, for example, it's not one gene, but you want to clone a cluster of genes. You want to clone a large DNA, and this DNA can be up to let's say 40 kb, 40 kb, right? So the length of the gene is roughly 1 to 2 kb, but when I'm talking about 40 kb, so in such cases you can use actually different type of vectors. When the length is up to 100 kb, then you are not going to use plasmid as vectors. Again, different type of vectors are used, and these vectors which can carry such large inserts, such large DNA, for example up to 100 kb or up to 80 kb, these vectors are called as high capacity vectors, because you can clone really large large fragment of DNA, right? The choice of vector also depends upon the host organism. So by this what I mean is that there are some plasmids which replicate in gram-negative bacteria, there are some plasmids which replicate in gram-positive bacteria. So the choice of vector would depend upon the choice of the host. You are going to select a specific type of plasmid which will replicate in that specific host. The third point is any modifications. So these modifications can be, for example, if the gene that you have cloned that transcribes and translates into a protein which requires some modifications. For example, any type of glycosylation, hydroxylation, methylation, phosphorylation. So in such case, a bacterial host is not a good host. So in such case, usually the choice of the host is mammalian cells. You can also use Saccharomyces. So in such cases, the choice of vectors are going to be different. Okay. So we will start with plasmids as vectors first, and I will not be going into the details of these high capacity vectors which are called PAC, BAC and YAC. You will learn these in 733. I'll be discussing with you the about plasmids and how plasmids can be used for gene cloning. So the discovery of plasmids actually dates back to 1952 when Joshua Lederberg introduced the term plasmid, and the name plasmid actually is derived from its location in the cytoplasm of the cell, and earlier in the earlier papers they used to call it as cytoplasmic chromatid, but then finally they combined the cytoplasmic chromatid into the word plasmid, and this is commonly used nowadays. The first plasmid that was identified was the E. coli fertility factor F, and why this is fertility factor and why it is named as F you will learn when I teach you conjugation. Essential features of a plasmid, so they are extra chromosomal, they are self-replicating, they are usually circular but sometimes linear. They are found in bacteria, but they are also found in yeast and certain fungi as well. The naming of the plasmids, so in the earlier days the plasmids were named based upon their function. So for example, F plasmid was named because of its function, which is fertility. R plasmid was named because this plasmid conferred resistance to antibiotics, and therefore the plasmid was named as R plasmid. Coli1 was named because it was producing colicin. So therefore these plasmids were named based upon the functions, the genes that they had and the different functions that they were performing, fertility, resistance or colicin E1 production. But later on, there was a scientist called Richard Novick. So he suggested a standard nomenclature for the naming of plasmids. So in the year 1976, he proposed that all plasmids, when we want to name a plasmid, the name of the plasmid should start with a small letter P. So whenever it's a small letter P, that means it's a plasmid. And then either you can write in uppercase the name of the university or the name of the research scientist. So for example, if you see plasmid PUB110, so here P depicts plasmid and UB is University of Bristol and 110 is 110th plasmid, right? Likewise, PSC101. So P depicts the plasmid, SC is the name of the scientist called Stanley Cohen, and 101 is 101th plasmid. So that is why the labs which are engaged in plasmid or bacterial genetics research, the moment a student joins the lab, the student is given a number that okay, you can number your plasmids from 1 to 100, something like this. So likewise, you can name your plasmid. So for example, if it is Deepak's plasmid, the name will be PDJ1, PDJ2, something like this. If it is Dhwani, then it will be PDT. So that is how the plasmid nomenclature is done. These days, the university names, for example, PIITD or something like that, this is usually not followed because when we write the university name, then there are many labs and that creates confusion. So therefore, plasmids are usually named based upon the initials of the research scientist. Then the size of the plasmid, so this I have already discussed in the class that it can be as small as 0.85 kb, that means less than 1 kb, which is from Thermotoca petrophylla, but it can be as large as 2090 kb from Ralstonia, or even 100 kb, which is the size of F plasmid from E. coli. So plasmids can really vary in size. And then the genetic composition of the plasmids. So plasmids carry two types of genes, backbone genes and accessory genes. So backbone genes are the genes which contribute to replication and maintenance of the plasmid, and accessory genes are the genes which are responsible for encoding some function which provides some added advantage to the host cell. For example, survivability under polluted environment, or for example under heavy metal contamination or antibiotic contamination. So in such cases, there are genes for antibiotic resistance, there are genes for heavy metal resistance, there are genes for degradation of polyaromatic compounds, or genes for degradation of any environmental pollutant. So all of these genes are called as accessory genes, and the genes which are responsible for replication and maintenance are called as backbone genes. So the plasmid has either both of these types of genes or only backbone genes. So we will be discussing about these in a little more detail in the next class, maybe on Friday, when I will be teaching you the classification of plasmids, which can be based on function, which can be based on copy number, which can be based on sizes, which can be based upon compatibility. So we are going to discuss these in a little more detail, and then from there, I will teach you how plasmids can be converted into vectors and how we can do gene cloning in plasmids. So maybe I'll stop here. If you have any questions, uh, please ask me.